Alright, this is Danielle Walker performing the experiment for chemistry lab number 5. It is called the titration for acetic acid and vinegar. The introduction. This experiment allows the students to use titration methods to define the concentration of an acetic acid solution. Titration is a method used to identify the precise amount of a solution that will react within or with given second solution of which the concentration is known. Students will be able to identify the end of titration by noting a color change once the acids and bases have neutralized. Students will also be able to determine the molarity, normality, and weight percentage of the solution. The hypothesis is that 5 milliliters will neutralize when mixed with a specific amount of NaOH. Okay, this experiment requires the use of distilled water, white paper, toothpicks and scissors. It also calls for the use of a 50 milliliter and 100 milliliter beaker, a 25 milliliter cylinder, a 10 milliliter titrator, a titrator stopcock, and a test tube clamp holder. The chemicals that are required for this experiment are phenolphthalein solution, sodium hydroxide, and vinegar. Students are required to set up the lab equipment according to the lab packet instructions and then drop by drop add NaOH to the vinegar solution, which is in here. And we will be able to observe and record the point at which the solution neutralizes. After repeating this experiment 3 times, we take the results and average them in order to draw our conclusion. So the procedure required that we No. Okay. So it requires that we put the test tube holder clamp, ouch, my knee, holding the syringe, and we place the 100 milliliter beaker underneath of it so that we can add the chemicals together at a regulated pace. The syringe is filled with NaOH. We added 2 drops of the phenolphthalein solution to the vinegar sample in the beaker. We only added 1 drop at a time. At 1 drop there was no change. At 2 drops there was no change. At 3 drops there was no change. 4 drops, 5 drops, no change. But at 6 drops, it began to turn a very slight pink color that almost disappeared after the solution had a chance to mix together. We did this experiment 2 additional times to note at which point the solution turned pink and remained pink after the solution had a chance to sit. Then we thoroughly washed and cleaned all of our equipment. And then the practical application of this experiment, pharmacists can use titration to mix compound drugs. Doctors can use the titration to decide the proper amounts of various medications that should be placed in an IV, to monitor blood glucose levels in a diabetic patient, as well as the use in pregnancy tests.